Hello, everybody. My name is Moises Agosto. I'm the Director of Treatment for NMAC. Today, we have a very exciting webinar and two excellent panelists, people that have dedicated a lot of their time, in the past decade or more on research related to AIDS cure. And we have them today, and we're excited and happy. And I'm very grateful that they agreed to be part of this webinar. Is this information that we really need, not only to share it with the community, but also we need to hear feedback from the community, as Linda and Jeff have been mentioning, as they belong to the DARE Community Advisor Board for the Martin Delaney Collaborative on HIV cure. So with that, I'm going to pass it to Linda to start with our webinar. Linda?
Thank you, Moises, my old friend. Yes, I'm Linda D, and I'm the Community Engagement Coordinator for the DARE CAB of the Martin Delaney Collaboratory. As Moises said, Jeff and I have been doing this work, I don't know, 35 years on my side and about that 30 or so on his side. And we've been working on cure for the last decade, I suppose, because we want to see the end of this. And we know that things are moving that way. I think we have to say in the beginning that this is not around the corner, and we don't want people to think that, okay, we'll be cured tomorrow, because it's not going to be that way. Science doesn't work that way. But let's jump in, and we're going to do an introduction to cure research. At the end, I'm going to talk specifically about some over 50 scientific issues that relate to analytic treatment or interruptions. I'll explain more of that later, and then Jeff will jump in the middle, and we'll be back and forth on the end. So, okay, so you can give me the next slide, please. So we're going to do a whirlwind tour of why we need an HIV cure. What are some of the ethical challenges? Why we believe a cure is possible? Why it's so difficult, and different cure strategies that are being pursued, which is probably one of the most interesting parts that Jeff will be talking about. Next, please. So what do we mean when we say cure? Does that mean that we're completely cured, that HIV is completely gone from someone's body? Or does it mean that HIV is controlled without daily drugs, translated to the ability to control HIV without your antiviral meds? Now, the thing of it is that obviously we'd love to have the complete cure, but probably this will happen in stages, just like antiviral therapy. We didn't get to one pill once a day overnight. So we'll probably have these interventions where you'll be stopping and starting, kind of like the long-acting drugs that we've been seeing recently, until we can actually get to something that's really complete. So there's two different ways of looking at it. Next slide, please. And what kind of cure do we want? Okay, so we have some requirements, and you can flip them all up if you want, because there's a number of them there. We want it to be safe. We have drugs now that are relatively safe at one pill once a day, which is pretty simple. I don't think they're affordable, but a lot of these other drugs, these other strategies are going to be a lot more affordable. So we want to be sure that we keep the prices down so that everybody gets a chance to get these drugs, not just rich people. That they're scalable. And what do I mean by that? We mean that our brothers and sisters in developing countries will also have an opportunity to have these drugs, and they won't be so complicated to make that it will be impossible for them to have access to them. We want it to be complete, what we talked about in the slide before, about being off drugs and eventually having it be gone from your body, and durable, which means for long periods of time. I mean, you don't want to have to take something once a month. We already have that, okay. And another big deal here is that, just like having a no viral load prevents transmission, we want that these cure drugs to have that same characteristic. Okay, next slide, please. And why do we need a cure? Okay, we can go to the next slide. I think the people that we've talked to, we've done a lot of surveys in the beginning of this endeavor, it would be liberating for both individuals and society. I mean, not only is it a drag to have to take drugs every day, but there's stigma that goes with it. There's just a lot of hassle and a lot of things that make your life really inconvenient. And every time you have to take a pill or deal with this inconvenience, it brings back to you that, oh, well, I've got this, possibly life-threatening disease. So I think that's where the liberating part comes in. Next slide, please. So here are the reasons why we need optimized treatments and a cure for HIV. There are about 38 million people with HIV across the world. And having drugs for 38 million people every day of their lives across the world is like 100% harder than it is for us to get drugs in the United States. And we cannot overcome these challenges either in this country or across the world for people's entire lives. There's drug resistance, which means all of a sudden you've been taking a drug and then it doesn't work anymore. There are numerous reasons for that. There are toxicities over time. Everybody's like, oh yeah, the kids nowadays will be like, okay, I only have to take 1 pill a day. But guess what? If you have HIV for 30 years, you're going to be taking 20 other drugs for other things that have come to you as a result of it. And again, as I said, the stigma that's associated with taking drugs every day. Next. So why do we need a cure? Well, when I just mentioned all of the drugs that you have to take for these different conditions, with HIV comes cardiovascular disease, which means heart attacks, premature aging and frailty. I think a lot of us know what that means. Weaker bones, neurologic disorders, cancer. Probably, you may have gotten a certain kind of cancer 10 years later in your life, but you're getting it earlier now because your immune system is flopped out. Diabetes, because maybe you have a propensity to that, but maybe the drugs are causing that to happen, sooner. Lipodystrophy, we've all seen people's faces that get sunken in, or humps and bumps that have been caused from the drugs. A lot of this is immune exhaustion, but we really haven't been able to tease out yet whether it's the drugs doing this, whether it's the HIV doing it, or if it's a combination of both. So it's a lot of ugly things in this slide that we would like to get rid of. Mainly that little devil in the middle, that chronic inflammation that makes pretty much every one of those worse. Okay, next, please. So why do we think a cure for HIV is possible? Next, please. What do we know that makes us think we can do this? So as of right now, there are 5 people who have been cured from HIV. Timothy Ray Brown, who maybe many of you have heard of, who was our little angel that was around for so many years and eventually ended up dying because his cancer came back, not because of his HIV. Adam Castillo, our friend, the London patient, has been cured. Mark, the Dusseldorf patient, has been cured for about 10 years, and he's now finally agreed to come out and talk about his life and his case. We have the New York patient who is not yet ready to talk about the circumstances, and the City of Hope patient, who is a lovely person who came out at the last IAS meeting. I think it's really the time before. They go in different cycles, but he's a lovely person too. So the thing of it is, though, that the first 3 of these patients were cured in a way that does not relate to most people. They had cancer, they had chemotherapy, they had for years, which didn't work, and they actually had stem cell transplants. And when the doctors transplanted new immune systems into their body, what they did was they transplanted them with CD4s that were essentially immune. And I don't want to get too deep into this, but that were immune to HIV. So your CD4 is the door to your immune system. HIV latches onto that like a lock and a key. It's locked in and flows up in your whole body very quickly and spreads, essentially is what I'm saying. So when their doctors realized, well, you know what, maybe we can cure them of HIV as well as cancer, and essentially give them this new immune system that was resistant, that that couldn't be infected by HIV because of what's called a CCR5, they actually have been cured. So we don't know if their cancer will come back, and certainly they've been through the mill, but they're actually cured. Now, what they went through, it could be life-threatening, the different procedures to cure that cancer with, the HIV cure attempt riding alongside saddle. So this is not for everybody. You don't want to put yourself at that risk of death when you could take one pill once a day. So it's really just for cure patients. Now, the New York patient was a little bit different than that, and so with the City of Hope. So, but they are way further back, or we are way further back in understanding those cases as opposed to these cancer patients. So it's just that we don't want you to think, again, a cure is around the corner, and right now it's only for people that had cancer for the most part. But if we can do it for them, that means we could do it for other people as well. Even though it might take us a long time, we're on the road, I think, to it. Next, please. And why is it so difficult? Is this you now, Jeff, coming up? Are we on 11? I think this is you.
Yep.
So I'm going to turn it over to my partner in crime, Jeff Taylor, who's going to do the next couple of slides.
Great, thanks, Linda. And a great setup. Move to the next slide if we could. So, Linda asked the question, why it's so hard to cure HIV? And the answer is the reservoir, where the HIV hides out. We all know that, if you take your meds, your HIV meds, you're undetectable, but that doesn't mean there's no virus in your body. It just means that it's not detectable in your blood. It's still hiding out in all these parts of your body, your brain, your lymph nodes, your stomach, bone marrow, and also the genital tract. So it's all over. And as we all know, if you stop your meds, the virus comes roaring back as if it, nothing had ever happened. So that's the problem that they're trying to solve with HIV cure research therapy. Next slide. So how are we trying to address this? What are we trying to do? Next slide. So a lot of different pathways. And the first is called latency reversing, which is just a fancy term of saying you're going to wake up the virus, right? This virus is latent or dormant or asleep. It's not doing anything in your cells because it's suppressed in the bloodstream by the HIV medications that people take. So the idea here is to wake that virus up and then find a way to kill it. So they call it kick and kill. Sounds a little violent, but depends on how you feel about your virus. But as we know, it's the cells, when they're in this latent stage or dormant in your body, they get incorporated or integrated into your own cells, or mostly your blood cells, but also other cells in the body. And so the idea is to activate or wake them up. And that way they release the HIV. And that way the body can recognize it, or the drugs you take that they give you can recognize it and kill it. And the cells will die, but so will the HIV. So that's one form. One of the downsides of this is they use latency reversing drugs in cancer a lot, and they can be really toxic. And the other downside is that they don't wake everyone up. And all it takes is one cell to be there. And once you stop medication, eventually that cell will wake up and start releasing HIV, and you're back to the races. It's started all over again, and you haven't really cured the HIV. Next slide. So the other approach is to use your body's own defenses. We all have antibodies to fight different infections, and we do have antibodies against HIV. The problem is that HIV is kind of a clever virus, and it mutates a lot and finds ways around antibodies, just like it did HIV drugs in the early days when we only had one or two. And so we had a cocktail that could stop it in various ways and keep it suppressed. So it's the same thing with the antibodies. But what they've discovered is that there were people who did a better job suppressing the virus, and they had special antibodies that they call broadly neutralizing, because they can control more kinds of virus. And now they're combining them, two BNAbs for 2 and TriNabs for 3. And so they're taking these, and what they do is they take these cells from people who are positive that work really well. They multiply them outside the body in a test tube, and then they put them back into people with HIV to see if the right combination of these broadly neutralizing antibodies can help suppress the virus. And so we've had some promise with this, and this may work in two different ways. It could be kind of an effective non-drug therapy for people living with HIV. So you would give them these, they might work for a while, and eventually you might have to give them some more because the antibodies don't reproduce in the body. So you're always having to introduce, just like with a drug, you have to give them new ones all the time. So unless they can find a way to get the body to produce its own antibodies, this will be, kind of a continual therapy. So it might be what they call a functional cure, where the virus is still in your body. You might have to take shots every year or so to make it work. But this is an approach that is being looked at both for therapy, for long-acting therapy, and possibly as a cure. So next slide. So, Linda was talking about the 5 people who have been cured. And so, as she said, what they did is they basically gave them a new immune system, right? And gave them new cells that knew how to fight, that weren't infectable, could not be infected by the HIV virus. So, obviously, as Linda said, that's not something anybody wants to do. That therapy can kill you, and it has killed some people. But they had failed their HIV, their cancer therapies had failed them, so they needed to do something. So it was a risk worth taking. But for the rest of us who are taking our meds and doing pretty well, that's not a risk. That's not an acceptable risk that anybody would want to take. So, as I always tell people, yeah, it's great for those 5 people, but kids, don't try this at home. It's not anything you want to go through. And if you talk to any of them, they will tell you the horror stories of going through chemo and radiation, more chemo for the bone marrow or stem cell transplant. They get sick with something called graft versus host disease that people with transplants get, because you're basically transplanting a new immune system into them. So there's a lot of problems with that approach. So they're trying to find a kinder and gentler way of doing this. And so what they're doing is trying to modify the cells or the genes themselves to make this happen. So there's a couple of ways to do this. I'll start with the one on the right, ex vivo, which just is a fancy way of saying outside the body. So what they do is they take cells from somebody living with HIV. They take out the T cells or stem cells from the bone marrow, and then they modify them in a test tube in the lab. And just like the people who had the transplants, they change those cells so that they cannot be infected by HIV. So they're basically immune to infection, and then they multiply those special modified cells and then put them back into the body. And the idea is that these will kind of incorporate themselves into your body, multiply on their own, and be resistant to HIV infection. So eventually, over time, these take over. HIV, once you stop your meds, HIV kills off any cells, your own cells that were modified, and it's the new cells that take over and form a new immune system for you. So that works really well, except that it's really complicated. The other idea that might make it simpler is to use viruses that we're all, around us all the time. Adenoviruses are one of the cold virus families. Lentiviruses include HIV that's been modified. So they use these viruses to incorporate this into your own immune system just through a shot. So they just give it to you. The viruses go in and create the modified cells and do the work for you. So eventually, if the approach on the right, doing it outside the body, works, they'll find a way to incorporate it into these harmless carrier viruses that could do the work for you, and you could just have a shot. And that's how we would make this scalable, because obviously the process on the right is not something that is scalable. You can do it, in a few cases in a small trial, but that's not how you're going to cure the 1.2 million people in the US, let alone the 38 million people worldwide. So something that's a simple one and done shot, or maybe 1 or 2 shots like the vaccines we get today, is hopefully the way that this would work. And down on the bottom, I won't go into it a lot, but we hear a lot of the news about things like CRISPR-Cas9, gene modification. They work like molecular scissors, basically, and just take your cells, cut out the parts that allow those cells to become infected by HIV, so they make them resistant to infection, and then those are what are allowed to multiply, either outside the body and re-injected, or used with a carrier virus to inject them into the body and multiply on their own and basically replace your immune system. So next slide. So another approach is to take advantage of what the virus already does. What it does is it gets into a cell and it relies, and most of the time, as we said earlier, it just kind of stays there, especially if there's not virus multiplying in the blood. So they have these long-term, they call it quiescent or senescent cells that basically stay asleep. So if you could just keep them that way, you won't have sickness. You won't have completely eliminated the virus from your body, but it won't do harm. And that's really, the next best thing, right? If we can do that, then we can call it a win, if it works effectively. And so the idea is to keep the cells from multiplying and reading the HIV DNA, because once that happens, the HIV takes over your own immune system cells, starts to multiply, and then causes infection, and that's what makes you sick. So if we can lock that DNA from ever being read, you've solved the problem, and people stay healthy without having to take HIV medications. So next slide. So, the question is, how do we make all this happen, and how do we do this research safely? So Linda's going to tell us about a meeting we had a few years back where activists and researchers and people from the FDA and other government agencies came together and how they made that happen. So Linda, take it away.
Thank you. So, we talked to Moises and Jonathan about bringing you some information from our Precure community workshop, which we have every year. And there really wasn't a lot of new information, but, there were a lot of people that were raising the mantra of, we want to be included in this research. So, wait a minute, I have to do my webcam there, cool. Yeah, we want to be included in this research. And why aren't we included? And why are the protocols that tell you who can come in and who can't come into a study, the inclusion-exclusion criteria, why does it exclude us? And, I couldn't help but think, sitting there, that, well, you know what, we need to really do some education about this, because every one of these trials, okay, at this point in time, that is actually trying to prove if an intervention works, requires that you go off of your medication, some for a short period of time until your virus rebounds, and others for a really long period of time, months at a time, to see if the interventions that they give you will either tap down the reservoir and bump up your immune system so that you can fight it off yourself. But, think of the consequences of that. Think of somebody who is over 50, who might have a lot of comorbidities. We all know that when you're over 50, everybody's at a higher risk for cancer. Why? Because your immune system starts flopping out. We all know that the guidelines, every guideline in the world about antiviral therapy says you have to take it, you can't stop. So here we are saying, okay, now it's time to stop your therapy. There are a lot of people that I think won't trust that, and think, okay, I'm not going to be a guinea pig here. But, really, when you get in these trials, that's what you're doing. So, I think that Jeff really feels the same way about this. It's not our job to get you to get into these trials. It's our job to let you know that they exist. It's our job to give you information and educational, information and where to find things and stuff like that. But it's up to you and your doctor to decide if these trials are right for you, okay? So, there are a lot of these trials. Just think of what Jeff just told you. That gene therapy stuff is high risk medication. You could die from that. You could die from the strategy that was used on cancer patients. To me, that kick and kill is ridiculous. If you've got virus, why wake it up? It's all over your body. So many bad things can happen. That doesn't make sense to me. I love the block and lock because And we have another patient that on her own cleared it from her own immune system because she had that deep latency that made the virus, it's kind of like herpes or zoster. It's in there, but, we can make it stay asleep. So to me, that makes better sense. But that probably, that block and lock, that strategy is the farthest behind, the least research has been done with that. So in other words, you need to have your eyes open when you decide you're going to get on one of these trials and stop your medication, after taking years to get your viral load to be suppressed, you're going to take the chance of it rebounding. Maybe it won't, maybe it'll come back, and maybe you won't be able to get it to be undetectable again. I mean, there's a lot of things. You're letting that inflammation loose. Maybe, this trial will last a year. Maybe 5 years from now, you'll get a heart attack that was caused by this that nobody can really prove. But there's so many risks and unknowns that people like Jeff and I, when we have talked to the doctors about ATIs, we've been very strict about what people these trials should be open to. Because, when you're a doc, you got to be gung-ho about your intervention and your protocol, and, you want everybody to come in, but that doesn't mean that it's safe for everybody. So it's our job as activists, to sometimes say, wait a minute, these are human beings and we need to be, the thing that the FDA's biggest role is safety and efficacy. So we want to be sure we do no harm. So at this Reagan meeting in 2018, we devised some criteria with a whole group of experts that for what it looks like, what these trials should look like, or the inclusion-exclusion criteria, what that should look like, what people can safely do these trials, which is even more become even more an issue, the safety part of it, in the era of COVID, because suppose you get COVID in between, then you're at a double risk of all of these horrible things happening to you. So anyway, and be advised, there's no, it's like being a lawyer. There's no, where's the law? What's the law? There are a lot of different trials that don't follow this exactly. This is probably the strictest criteria, but there's some that may be 450 CD4s or a little bit different. But this is what came out of the actual first meeting where this was discussed. So first, to be included, you have to have a stable CD4 count greater 500 or greater. Your viral load has to be undetectable, and you have to be stable on your medication. And you have to be otherwise healthy without major comorbidities. So what we have, most trials cut people off at 65 just because it's safer, and mostly for the drug company it's safer because they don't want their drug to look bad. So what we have said to investigators is, okay, let's cut it off at 60 unless people have, and can I have the next slide, please, any of these comorbidities that are coming up. Okay, so, some of these hopefully, or unfortunately, should I say, will be familiar to people on the call if you're over 50. So in the exclusion criteria, and again, maybe if you're 65 to 70 and you don't have any of these, you could be eligible, but again, safety first. So if you have active or chronic hepatitis B, C, or active TB, no way. KS, Kaposi sarcoma, lymphoma, or other virus-associated cancers. Why would you want to wake that stuff up, okay? If you have it or if you have a history of it. If you have a history of dementia or PML, which is a great big long word for a horrible brain disease for which there's no drugs, no treatment that can kill you in a very horrible way. Because if the HIV gets to go run wild in your brain again and you can't get it controlled, you're in danger of those two things. You can become resistant to, or if you have resistance to 2 or more classes of medications, because when you go off your drugs, maybe usually it's ineffective drugs that gives you resistance. But in other words, we don't want to take people who have no other options and then take them off their drugs and hope for the best. Again, the other cardiovascular events are high risk of an event for heart attacks or anything that, AFib and a million other things that are related to your cardiovascular system. A history of age-defining illness according to the CDC. We don't want any of that to come back. This one we didn't get through, but I think is still important. A history of a CD4 nadir less than 200. And what does that mean? In other words, if your lowest viral load, your lowest CD4 count was ever 200 or below, nobody knows exactly, but everybody thinks, it hasn't been proven, but everybody thinks those CD4 cells are not as strong, your immune system is not as resilient, and that there's something about getting that low that makes your body and your immune system different. If you're pregnant or breastfeeding, if you have advanced non-alcoholic fatty liver disease or non-alcoholic hepatitis, that NASH, thing of it is there's measures for all this stuff too. Kidney disease, I left all that out, but I'm just giving you a flavor. And let me finish the last one. Children younger than 2 years of age at the time of the ATI. They have their immune systems are nowhere near, ready for prime time. So you can see why we would want to exclude people like this. And if you have any of these conditions, and again, a lot of these have different numbers that a doctor will look at to see if you might be safe. But if you're on the borderline, or if you have high numbers in any of these categories or low, whichever is the relevant metric, you don't want to get near these studies. You could, it's up to you, it's your choice. But again, we think it's our role to let you know what's risky and what's not. And this is the reason why a lot of people over 50 or 60 are excluded from these trials, because why? They have to get off their medication. And any of these categories in your life could really F you up. Let me just put it to you as straightforward as I can do. So, all right, there's that. Now, let's see. The next slide is.
Can I add a few things to that, Linda?
Yes, sure. And I was going to say, it might be a good place to stop here. We're only at 3:30 to ask people if they have questions before I get to this last advocacy slide. So jump in, Jeff, please, and then we can take some questions.
Yeah, I just wanted to let people know, this is a scary list of things. And, most of us, this is a group of, older people, living with HIV, long-term survivors. This is almost all of us, right? Which means that most of us would be excluded from these trials. But it's important to remember that these are for phase 1, first in human trials, the riskiest of the riskiest studies. So as Linda said, it's safety first. If somebody gets into these trials and they have, their TB comes back, or a cancer happens, or they have a heart attack, how do you tell if that was from the treatment or from something they already had? So it's really important to exclude those people so it's not, messing up the results. You want as clean a population as possible. That said, I think, all of us, Linda, myself, and other, activists in the field really want to make sure that the people who enroll these trials look like the people who are living with HIV. And these days, half of us are already more than half are over 50, and that's just going to keep growing. If any of these things work, it's going to be probably 10 years at a minimum before they actually get approved and become accessible. So, people are going to be even older, 60 and 70 and above. So we need to make sure that we study it in this population, but do it safely. And so one of the things we've been telling people is, any exclusion you have in a study, you have to have a scientific rationale. And the age is the biggest sticking point. They just automatically say, 50 or 60, and they just throw a number out. It's arbitrary. And we're saying, no, you have to have this list of things that you're concerned about. And those are the valid scientific criteria that should decide what the exclusions are, not just picking a number and saying, well, if you're older, you might have more problems. Because we all know people who are, perfectly healthy at 65, who have none of these things, who could be in these trials. And you could have somebody who's, 60 or 59, who has a lot of these things. So it's really important to make the science determine who gets in and who doesn't in safety. And then once we get, one of these treatments, these approaches that we've talked about looks promising, then you go into phase 2 where you have larger groups of people. That's when you start to loosen up these criteria and make sure that the study population looks like the population of people who are going to end up taking these drugs. So I just want to kind of put that out there to put it in context for folks.
Yeah, no, that's perfect. Very good. So do we have any questions about what we've talked about so far? Who wants to do the, are we raising hands, or what's the process? Moises?
So, hey, this is Chip. So we have questions in the chat box. We asked everybody to put their questions into the question box. So we do have several.
Okay, why don't you read
First,
it to us, Jeff?
so I'll read them off
In
to
other
you.
words, say it. I mean, I don't mean shoot it to us for the chat. Just
Yes.
ugh.
So let's go with a couple here. Who do I talk to if I have questions about a trial on HIV cure research?
Well, that's a good question. You know, let's switch to the last slide just for some context. So there is something called the Martin Delaney Collaboratory that are funded sites to do this cure research. So if you happen to be in Philadelphia, Chapel Hill, North Carolina, San Francisco, Atlanta, Boston, Baltimore is for children or adolescents and infants. La Jolla, which really covers the whole pretty much of LA, San Diego, or New York, you know, there are people that you can call, you can get in touch with right there. Now, if you know, you can see what trials are available, because of course most of these sites are not doing any clinical research. They're doing the preclinical stuff. But you know, I think they could get you in touch with, and there's community people, there's community advisory boards for each one of these things. I mean, I'm going to give you a link of how to reach these people at these different the community sites so they can get you started. The treatment, is that me? No. The Treatment Action Group on their website. I mean, Richard Jeffries does a great job of letting people know what cure studies are available, you know, with information about each one. I mean, if you're a computer geek, you can go on clinicaltrials.gov, put in cure, and they'll tell you what cure research is available and where, you know, what stage it is, what phase it's in. Phase 1 is first time in humans, as Jeff said. Phase 2 is, do we have a chance of this working? Phase 3 is, wow, let's give it to more people and see what happens. But most of these trials at this point are stage 1 and 2, and they have just a very few people in them. So hopefully that's helpful.
Okay, and we have several more questions. Jeff, did you want to follow up on that?
Yeah, well, you know, the other person you should talk to is your doctor, right? And then also your partner, because a lot of these drugs, as we said, these trials make you go off your medication. So an important consideration is at some point you may become HIV positive again. You may become viremic. You have virus in your blood. You could pass the virus on to your sexual partners. U equals U wouldn't work in that case. So it's really important to think about that. And if you have partners or if you have, you know, anonymous partners, to think about how you navigate that. You know, are you willing to use condoms again? That sort of thing. So it's not just you, right, that's impacted by this trial. And it's a whole other can of worms. So we could do a whole other webinar on that, but I think it's really important to let people know that that could happen.
Yeah.
Thanks.
Jonathan,
Okay.
I sent you a link for the chat about how to get in touch with each one of these trial sites. So maybe you could put that in the link in the chat if you can find that link.
Next question I have here, are there any trials for anyone born with HIV?
Very interesting. Jeff, since you're working on that, you should take that question.
Yes, most of these studies don't have limit people by, you know, if they're under 60. So they would absolutely qualify. And I think we need to make sure that when we start doing the bigger trials of phase 2 and phase 3, as Linda was talking about, to make sure that we enroll people from all these different subpopulations. Obviously, we need a good mix of men and women, trans folks, people of varying ethnicities, older people, and younger people, but definitely somebody who's been born with HIV, has been living with HIV 30, 40 years now, just like the old folks, right? So, but they, you know, were going through this when they were, you know, during their formative years. So it might look different. So it's really important to do that. But no, to my knowledge, none of these, most of these studies are people, you know, they want to be adults so they can give informed consent, right? So 18 to, you know, 60, 65, whatever the upper age limit is. So absolutely they're included. But I think you raise a good point that it's important to make sure we enroll enough of these people that we can see if there's a difference in them compared to people who have been got HIV later in life.
And that was actually one of the main points that we talked about at CROI was how to outreach to people that that are called long-term survivors because, you know, people over 50 are long-term survivors too, but these people are not over 50, you know, 50 and over. So it is very
Also a shout out to NMAC because they have a group. They've been, working with this population. This is a verticals group led by Portia Dees, who's like born with HIV and is a long-term survivor. So there are groups out there. So yeah, talk to the folks at NMAC if you want to get in touch with them, and they can hook you up.
Okay, Chip, next.
1 second here. Let me just scroll through. Isn't a CD4 count less than 200 considered an AIDS diagnosis, and isn't that a permanent diagnosis?
No, I mean, you know, in other words, back in the day, before there was this very effective antiviral cocktail, I mean, people used to have 5 and 6 CD4s, and they named them and every other thing. I'm sure there are some of you in this group that are familiar with that. But, you know, in other words, once you're below 200, okay, that is AIDS defining. But then there have been people that have been, there are people to this day that present, you know, to the hospital that are just finding out that they have the HIV and their T cells are very low. But then they take medications and they often go up. You know, I mean, just think of how many lives were saved when that effective cocktail came to be. You know, there were people that were on their deathbeds that actually are still alive today.
Yeah, and the questioner is right. Once you get that diagnosis, it technically never goes away, even though, I had 2 T cells, 30 years ago, and now I've got, 800. So, and, there are a lot of us like that out there. But the concern is, and that's why you saw that exclusion on the list of people they were going to keep out of those early trials, is that often when people stop their HIV medications, they go back down to their lowest point.
Yes.
And they're afraid that might happen. And anything below 200 would, of course, be, really dangerous. They're afraid that somebody who had that really low T cell back in the days, that could happen more quickly, and that might not catch them quickly enough. But most of these studies have very stringent restarting criteria. So they try to monitor you as often as possible. And that's another issue, is how do you do that without having to go to the clinic every week to make sure that they haven't, the virus hasn't come back, and that their T cells aren't starting to go down, because they want to catch people before they get to that point, well above 200, so they're not in danger of getting opportunistic infections that we all remember killed so many people back in the day.
As I said too, that was the one thing that we didn't get through in that Raygon criteria, but that we really believe is extremely important, because your immune system is different once you get that low, we think. And we just couldn't get it through. And some of the trials say a dual, say 350 nadir, or something like that, but most of them don't say anything about it. And I think that's unfortunate.
That's
okay.
why you should talk to your doctor. Sorry, go ahead.
Sorry to that. We have one more question. Is there any limitation on age to enroll in a cure trial?
Yeah, so that's the kind of examples that we were making. That usually clinical trials stop all clinical trials. Most all, you can't say all, everything, always, never. But most trials, the cutoff age is 65. Now, we try, there were some people that thought it should be 60 for cure trials because of, you want to flip back again to that slide right before this, because of all of these possible, diseases, conditions that people could have. In other words, what we're saying, and I think what Jeff explained before is that, okay, we're not going to go down to 60. We're going to stay with the norm of 65. And we're even going to go up to 70 if you don't have any of this stuff, But if you do, it's not a safe trial for you to be in. So, and they're all different. Most of them will probably say 65. Anything you want to add,
Okay.
Jeff?
No, any other questions? If not, we can kind of talk about how people can, more about how people can get involved.
Right.
We have one more question here. How how do you know if you have the deep virus?
How do you know if you have what?
Says, how do you know if you have the deep virus?
Oh, well, you could go to Boston and let them let them take a lot of blood and find out for you. I mean, there are the person that we know who is out in the, she's the San Francisco patient, who was cured of this on her own without ever taking any medication, without ever doing anything but going to Raygon and getting blood draws. And they found out that she was able to clear HIV from her own body, her own immune system. And it's because they're calling that the deep latency. So, in other words, how would you know that? You'd have to maybe you've been sick, have been positive for a really long time and never had to take drugs. Your CD4s have never gone lower than 500. You've never been sick. You'd be a real outlier from the normal person. So I hate to be so all over the place with it, but there's not many people like that around. And the point is, we want to try and make more people like that so that this sleeping monster stays asleep as opposed to waking up and wreaking havoc. Jeff, anything to add?
No, there's a recent case called the Esperanza patient, who's another example of that. So, and interestingly, most of them are women, which I think shows us that women really are the stronger sex. They have better.
How about it? And there's been a biracial patient, which is good news. I mean, it's funny. Some people don't want us to talk about that, and other people say, well, we're glad that it works in both races. You know what I mean? So, I mean, it's a double-edged sword there. So hopefully I didn't offend anybody, but I think it's important to note that this has gone, it's not just white people that we're talking about here.
Yeah, and I think you're right. It is important to talk about race because the people who are naturally immune to HIV, there's a very small, maybe 2-3%, are typically Northern European. So the concern was that you had to be able to get a transplanted cells from somebody who was from Northern European, and you had to be that way yourself so you wouldn't reject them. You have to have a close genetic match, right? And so what was unique about that one was, this person was biracial, and they used 2 different kinds of stem cells. One was from a closely matched person, I think it was a relative, and the other were stem cells from the umbilical cord blood from babies. And these cells are much more adaptable, and you are less likely to have an immune response against them or a rejection reaction. So that's kind of the way the future is, that we can use these banked cord blood samples from babies, and that can make sure that anybody could benefit from this kind of a transplant treatment.
So can we go to that last slide again, and maybe we can talk about, maybe it would be a good idea to tell you what we do, and you could decide if this might be something that you want to get involved in. I mean, Jeff and I both are long time, we've been giving presentations about treatment in the early days, all of the research, then once drugs were approved, and then prevention, and now this. So we're old hands at doing this. And you could actually learn this stuff and give presentations yourself. You could help a community-based organization in your vicinity that's interested in doing these things. I mean, Jeff and I, we like take the show on the road from our own offices, right, Jeff? So we'd be happy to talk to your community about this. You could help organize a meeting like that. The other thing that we do is we were both CAB co-chairs, and we're now both community engagement coordinators, which means that we actually do these events like we're doing today to make sure we get the word out. I mean, for me, it's extremely important. I mean, we've watched, everybody, only white people get all this information, not purposely, but that's just the way it is. And, we're very, passionate about making sure that this information, this cure information, gets out to everybody, just like we've been about PrEP, because just for instance, I mean, the communities that are the most hard hit are African American, MSM, and trans people. So, if we're going to have a cure, we want everybody to know about it. So you can help get the word out in your community by getting in touch with people like us about maybe doing something like this. I mean, in our own groups, with me with DARE in San Francisco, I'm from Baltimore, but that DARE is who I work with. Jeff in Southern California with REACH and also with DARE. I mean, we deal with the administrative stuff of making the trains run, of getting this stuff done, of making things, getting the money from the investigators, of dealing with other community advisory boards around the country, of just being educated about this kind of stuff, so you can be on one of these community advisory boards, so you can have meaningful input on what a trial should or shouldn't look like. We're doing a lot of papers about what risk is too much. What people. I think the point of our next thing is going to be our aging, our 50 plus long-term survivors. Is let's do some surveys. Let's get some opinions about the risk that 50 and over and long-term survivors are interested in. What is most important to them? Something that's really important that we all need to think about, I think, that is not immediately apparent, but it's immediately apparent every time we spend a lot of time. And Jeff's been with me. I mean, we've been at San Francisco AIDS Foundation and done these talks, and people all of a sudden they're like, oh my God, I won't be HIV positive. And all of a sudden, their 30 years of identity goes out the window, and the people that they're friends with, and the support groups that keep them alive, and the money to pay for their drugs, and the money to pay for their health care, all of a sudden is gone. So we're really trying to be the canaries in the coal mine with the researchers to say, okay, if we get people cured, hallelujah, baby. But guess what? Where are they going to get their medication for all of that list of stuff that they may have wrong with them, because either they've been infected for a very long time, or they've, that need to take other medication for other things that are caused by the medications for HIV.
Yeah, and that's a really important point. You hear that a lot. So as part of the advocacy we have to do, not just with researchers, but also, with Ryan White and things like that, to make sure these programs don't go away, and we have a way to kind of reintegrate people back into the general healthcare system, back into work and things. But realize that, as Linda was saying, you cure the HIV, but you can't cure everything that happened to them in the course of 30, 40,
Right.
50 years by the time we have one. And then the other thing I wanted to mention is
the ACTG, right?
Yeah, exactly. So I just
Okay.
wanted to talk about that, and I just shared the contact information in the chat there. But as Linda was saying, these Martin Delaney collaboratories, they do the really basic preclinical research. So we're talking test tubes, looking at mice and monkeys. Most of them are not going to do human trials. That's going to happen at a group called the AIDS Clinical Trials Network, or the ACTG, and they've been around for 35 years now. And that's where all the pivotal trials, for the first drugs and so forth happened. They're the ones who do the big clinical trials in large groups of people. So when we have, a cure approach that looks like it's going to work, they're going to be the ones to do those larger trials, enrolling as diverse a population as possible. So they have a very robust community presence. Back in the day, the AIDS activists and TAG, they fought their way into those meetings. They were closed. And they said, oh no, nothing about us without us. We're going to be at the table, and got them there. And so to their credit, they have a community advisory board at every site all over the world. It's a global network now. So all over Asia, Sub-Saharan Africa, South America. So it's a really diverse group. And so that's where these things happen. And anybody can join if you live in any city, even if you don't, where they have, now that everything is online and on Zoom, you can join a local CAB meeting, and most larger cities have them. And then they also have something called a community scientific subcommittee. And these are the people who are, have more time to give and are really invested in making sure research gets done right and ethically. And so they sit on the protocol teams, they help design the protocols. They're the ones who make the decisions about funding and so forth. So community is at the table and helping guide the process. And so that's really where you can make a difference, and also a great place to learn. So you can take that information back to your communities, because that's one of the missions of the community advisory boards, just like what Linda and I do at the Delaney Collaboratories, the same thing. They have seminars and invite people to their meetings to make sure the community knows what's going on. And when the trials are open and ready to enroll, that it's happening. so that we can get as many people in them and get, the right people in them, make sure it represents the people being affected by the pandemic. So I've shared that information. I don't know if you're able to put that in the chat or not, but we can share that afterwards as well. But just Google actgnetwork.org. And there's a lovely woman named Allegra Cermak who started out, at the very beginning as one of the community people and is now their network coordinator for the community.
That's where we learned 90% of what we know.
Yeah, it's a great place to learn how research gets done.
Jonathan, I looked at my emails to you, but I know I sent you that link for the Delaney Collaboratories, how you can get in touch with each one of these organizations. So if you maybe want to search your email and stick it in there real quick in the last couple of minutes, that would be helpful, I think. How about other questions? Do you think that did you learn anything? Was it what you expected? Was it helpful? Was it completely useless? The talk today, how do we improve it?
We've had a couple of people asking about getting in touch with you. Is it okay if we share your email addresses with everyone?
Yes.
Please.
Okay, Linda, what's yours again?
It is Linda, Lynda, M for Marie, DEE at AOL. That's how old I am.
I used to have AOL.
Right.
Mine is jeff.taylor@harp, HARP -PS, like Palm Springs.org.
I should probably give you, what, you should probably use the AIDS Action Baltimore one. That would, instead of that old personal one, use Linda D, LYNDA at AIDSActionBaltimore.org. It'd probably be better. Gmail wouldn't block you.
Let's see, a few more
Do you
here.
have any
Let's
comments
see.
about the critiques or that sort of information?
People are getting there. Let's see. Let's see. Lots of thank yous. Lots of requests. The website's in the chat box. It's actnetworks.org. Some questions about info in Spanish for monolingual people.
Yeah. Well, we've been talking about. I mean, we did with Jeff actually did one session in Spanish, and we were talking about doing a Spanish speaking webinar series after the aging one, but it's probably going to be a good 6 or 9 months. And we actually have done a lot of stuff for San Francisco AIDS Foundation for their support group, but are people that are English is not their first language, but neither one of us speaks Spanish. So maybe Moises could offer some tips on what we could do about that.
Well, like Linda said, we did do a webinar in Spanish. Adam Castillejo, the so-called London patient, is actually originally from Venezuela and a native Spanish speaker. So we did an interview of him with a Spanish researcher in Vicente Planellas, and that's on our HARP PS website. So if you go to YouTube, it's on our website, but also on our YouTube channel, and look up the HIV and Aging Research Project, it's there, and I can look up the link and send it to them. But yeah, it's an interview in Spanish talking about what he went through as one of the people cured of HIV. It's a fascinating story. And like Linda said, we're planning to do a whole series going forward. We've already got one planned with some researchers in Barcelona.
That's a great action item for next time, and I think Moises will be really excited to do that. So I just want to respect time, and we're at that hour. So thank you both, Jeff and Linda, for sharing so much great information. This crash course. I know everybody got something out of it. So thank you both so much. We really appreciate it.
You're welcome.
And thanks everybody for joining us. We will be sending out some more information about our next webinar. So this is the first of 5 for this year. So stay tuned for that. And thanks so much.
How many people we have? 58?
Yeah. More.
Good. Thank you everybody for coming.
Thanks all.